All right, welcome everyone to another edition of Omega Matters. Today, me, Kristina, and Bill, Dr. Harris, are just going to chat a little bit about a research organization that we're both a part of called FARI, the Fatty Acid Research Institute. And if you don't know about FARI, that's not surprising because it's pretty new. And they're just getting going. So we wanted to talk about that, why it got started and what kind of work they're doing. So that's what we're going to talk about today.
Great.
Sounds good. You want to start up a little PowerPoint?
Yeah, let me do that. Let me see, share my screen, and that's the screen I want to share. And here we go. Yeah, can you see that? Okay.
So FARI, why did FARI start?
FARI got started partly because I guess the honest reason is OmegaQuant, the laboratory, of course, we're both part of, for some years was kind of funding research. And that really, the research that was not specific to OmegaQuant,
right,
per se, was specific to the omega-3 fatty acid field. But as a laboratory, as a company, that's really not part of what we need to be spending our money on. So the idea, I think, was, well, I'm going to spin off because I, as primarily a scientist, founded OmegaQuant, back in 2009, but have never really been all that enamored with the business side of it. It's okay, you learn things, but that was not my sweet spot, and that was not my particular talent.
Yeah.
Research is what I've always enjoyed, and so the opportunity to get back to research and lead a nonprofit group that would be eligible to receive research funding, which OmegaQuant as a company really can't.
Yeah. Although
led to the idea of starting the Fatty Acid Research Institute. And so we did in 2020.
Yeah.
Little known fact, do you know what that logo is? A little dot on the eye.
It's not a windmill.
No, it's not a windmill. And it's not a Korean flag or
a Korean.
It is it's a very subtle, lost on most people, mark, because there is a bond going to the R for FARI. And this is the R group in organic chemistry. And then this is a carbon and a alcohol and a ketone. So this is a fatty acid with pretty, deep. I know.
That's a deep cut.
Deep stuff. Yeah. Well, nobody's
asked.
Yeah, now here we are. Anyway.
But that's good. Everything is intentional.
There's a reason for everything. Exactly. So that's why we started FARI, for that kind of, in a way, commercial reason, but plus to get me more back into the research area. And also the higher goal, the more altruistic goal, the reason for a nonprofit research education group is to advance the science of fatty acids and health, with a particular emphasis on omega-3, but not exclusive. Because we think there's a lot of information in fatty acid profiles, whether it's red blood cell or plasma or whatever,
right,
that needs to be dug up and exposed and brought into practical use if possible. So that's kind of why we started it, and it's been going now for about 2 years. And so like that was kind of my, let me see if I can advance this. Should be able to. Well, of course, on the other hand, maybe there we go. So this was all, that's what I just said.
Yeah.
Say it again. We have a group of scientists now involved with FARI. Very, very happy to have this group. We have people we have 3 physicians, Dr. O'Keefe, Dr. Wallace, and Dr. Marchioli. And we have Dr. Park here, who is a professor in Korea of nutrition, an old postdoc of mine. Nathan Tintle is our lead biostatistician and our executive director. Plays
grants
major
manager, and
grant manager, the whole thing. He does everything.
he does.
Michael McBurney is in Toronto, a nutrition scientist. Jason Westra works is a biostatistician, works with Nathan.
Mhm.
Because biostatistics is what we really do.
Yeah.
We've got Alex Salavila, who's a nutrition scientist in Barcelona. And of course, you, Dr.
Yeah.
Jackson,
There it is.
Brings our specialty of early child development and pregnancy to the fore here. Recently, Rhonda Patrick, who is a very broad-based, she's a podcaster, foundmyfitness.com, very, very good podcast she does, a very wide range, but very interested in omega-3 fatty acids. And Rhonda's bringing some specialty in the cognitive area. And so we're excited to have her on board. And then Jean-Philippe Schuhart from University of Hanover in Germany. He's a professor there. He's got several projects of interest and students he's working with. So that's our group. And we're looking to expand. The general idea is we make data available to researchers who want to write up studies. They ask the question, we do the data, and we give them the answer.
And so it's just interesting to see all these people together, and they're obviously basically everybody is pretty far, like terminal degrees kind of thing, but it's from a lot of different areas and a lot of different perspectives. And
Right.
fatty acids is the main thread, but all over the world, all kinds of different interests that do center around fatty acids, which is really cool. And starting this in 2020, it just lent itself to online virtual collaborations.
Exactly. In the thick of COVID.
Think of COVID, and like this just made sense. And it's also just a kind of a different way. Research is kind of a tricky field. You write a grant, you write lots of grants, you try and make that grant make the funder interested in it, but it may not be exactly what you want to look at. And for this, we'll talk about it, but lots of data, lots of big questions, and we still have to find funding, still have to do grant writing, but there's a little bit more just openness, it seems like, to questions you can ask.
And it's not nearly as expensive,
right,
to do the research we do, because we do generally work with existing data.
Right.
It still costs money. I mean, biostatistical time is valuable time, and that's the crux of the whole thing.
Yeah, it's kind of the big data revolution.
It's sort of the big data world, right?
And it is connected to force. I don't know if you're going to talk about that here.
Not specifically. I'll mention a paper or two. Peripherally related to the Fatty Acid Outcomes Research Coalition from Tufts University, where we participate very deeply with multiple studies that they do. But that's not.
It's a Venn diagram.
It's kind of a Venn diagram of how we work together, right? Exactly.
Nice.
So I mentioned cohorts, and this is let's see if I can pop down this little thing. Here is a list pretty complete of the cohorts. Cohort, of course, is a group of people who are being studied in a systematic way. The classic one here is the first one on the list, Framingham. It's Framingham Heart Study. The offspring is the second round of Framingham research. That's the children of the original cohort. Original cohort was assembled in 1948 or so, and these people were recruited here roughly in the 1970s. There is now, to skip down a row, there's Framingham Generation 3. And that's the now grandchildren and spouses of the original cohort. And so that's, we have blood analyzed, and all these we have analyzed, red blood cells or whole blood, and for fatty acid composition. And then we're following them. So for example, in the Framingham cohort, it says here, the blood was drawn that we analyzed the omega-3s in, the fatty acids between 2005, 2008. The people were about 66 years old at the time. There were around 2,900 of them. They were generally healthy, drawn from the community. Of course, everybody's got some disease eventually,
Right.
but they weren't picked to be a particular disease. And the amount of follow-up we have at this point is roughly 17 years of time for different diseases to develop. And the plus simply means it's ongoing. There will be 18, 20, 25 years of follow-up eventually as time goes on. And different diseases will develop. So that plays into this. A much smaller cohort, Omni is one of the Framingham. It's only 300 people, but they're non-white. The Framingham cohort is almost all white. So this is an effort to be a little more racially diverse in Framingham. Generation 3, the same thing. It's Omni 2. That's a non-white cohort that matches up with the Gen 3 cohort. Then go ahead. Do you have a question?
Just to to clarify, every year the medical records are updated and kind of cleaned up by the Framingham group, and then different research organizations can apply to get that new wave of data. So like the data is always kind of changing as
It's always being
endpoints are being brought in, but you still only have one blood time point that you're mostly working on.
Yeah, it would be very good to be able to get funding, back to funding, to be able to analyze red blood cells, say in the Framingham offspring at a exam 10 years later.
Yeah. That'd be great.
And then to know how stable omega-3 levels are, because we are presuming in all these studies, anybody who does fatty acid based or biomarker based epidemiology presumes that the level in the blood at the time it was drawn remains that level over the time of the follow-up. And that's generally true. We've had one paper in Framingham where we've supported that. Although we did show that if we specifically on the omega-3s, if you measure someone's omega-3 level at one time point and then a follow-up like 7 years later, and they have started taking fish oil pills, surprise, surprise, their omega-3 levels go up.
Yeah.
So we can't, you can't presume unless you know somebody started fish oil that their levels are going to be the same. Anyway, follow-up would be helpful.
And in the follow-up for the clinical records, do they have anything with diet, or if they take any supplements, is that a covariate you can use?
Yes, they usually have updated records of drugs being used, supplements being used, and then they do dietary surveys. So it's possible to, for example, we wouldn't know the omega-3 level of a given person at a later follow-up time if they started fish oil supplementation. We could guess at it because we know it's about 2 omega-3 units higher.
Right. But that's assuming someone can say they're taking a supplement and it's like once a week, or I did it for 3 months and now I'm stopped. So there's issues with all kinds of data, but it's interesting to get into the weeds a little bit of what these data sets are good for and aren't good for. And one of the biggest benefits is just how many people are involved. Those numbers are huge. You could never do RCT, randomized controlled trials, with these numbers for all of the different endpoints that you guys are looking at. Well,
or over the years of follow-up. You couldn't do a randomized trial that lasts 17 years.
No, it's not possible.
It's not, it doesn't happen.
And you're looking at hard endpoints. You're not just looking at necessarily, is this biomarker and that is a biomarker changing, and that biomarker has the risk for heart disease or something, because that is another variable. Like when you have the intermediate, when you're looking at biomarker changes instead of hard endpoints.
Oh yeah,
a little bit lower quality. So high number of people, as you know, they're not all in the same place. So you have some geographic diversity, long follow-up times, and actual medical record hard endpoints like diabetes or heart disease clinically diagnosed. It's pretty powerful for that.
It's pretty powerful for that.
Yeah.
Exactly. Just quickly, Women's Health Initiative Memory Study. It was another study. We have about 7,000 red blood cell samples analyzed from, but these were healthy postmenopausal women. Again, sampled in the late 90s. So it's been some time for them. We've got some interesting studies out of them. Puerto Rican Health study is done in Boston, hence the name. And we really haven't done much with them yet, but we have access to their data, and we've analyzed their red blood cells, so we trust them. UK Biobank is a unique one here because we at OmegaQuant haven't measured red blood cell omega-3 directly in the UK Biobank. That's been measured by a different analytical method, and we've correlated our omega-3 index with it, which opens up the door to the address. You see a huge sample size. It's 117,000 people. Roughly sampled about 14 years ago. By the end of this year, they should have everybody analyzed by their what's called NMR. It's the method they use to analyze fatty acids and a lot of things in the blood. And it'll be almost 450,000 people that will have blood omega-3 data on, and so it'll be a very happy hunting ground for looking for correlations with disease outcomes. Vitals, a randomized trial. We were able to analyze about 1,000 samples in that one. It was pre and post omega-3. And so we'll be hopefully doing some studies there. Predetermined was another observational cohort of 6,000 people, but these were individuals who had a history of a heart attack, and they have a low, poor ventricular function. They're kind of on the edge of heart failure. So it's a unique population we haven't really dug into yet, but we've analyzed the blood for them. So there are outcomes there to be studied. And Strength finally is a randomized controlled trial like Vital was. And we have samples on about 13,000 people. We know, again, red blood cells and plasma, interestingly. But this is only a 5-year follow-up, just like Vital, because it was a randomized trial and it stopped. So they haven't continued to follow these people after the study was over. So that's an example, or that's pretty much the total of the kind of fatty acid groups for data we can get at.
And the biobank looks like they'll be analyzing and including a lot more samples by the end of this year. 40.
Right. This thing, what it is right now, and they intend to have about 500,000 done, 450,000 done by the end of the year. So it's a great research opportunity there. So here's kind of how it works, how the FARI approach will take it. One of our scientists says, I wonder whether blood omega-6 levels predict risk for knee transfer or knee surgery, for example, just an outcome of some sort. The idea is proposed, and then a kind of analytical plan is really developed in getting specific which cohort, what outcome exactly, what blood measures are available. Then Nathan and Jason, our biostatisticians, will assemble a database from the particular cohort that's being studied. They will number crunch, which is easy to say, but it's much more complicated than that.
Yeah.
And then provide the results basically back to the leads, whoever proposed the idea, and that person then is responsible for writing up the introduction, writing the discussion, interpreting the findings. So and then submitting it to paper and shepherding it through the publication process.
Yeah. Yeah. So it's nice to have these statistics separated from the person writing the paper who may.
Oh, yeah.
That's always a helpful process. And these, like, analyzing fatty acids and really like any human biological data, it's not as cut and dry as just regular old statistics. Biostatistics has a little art and a little science to it. So
oh, yeah.
Nathan and Justin have so much experience with this kind of data that it just makes it go so much faster.
Right. Yeah. Biostatisticians theoretically can work with any numbers.
Right.
but each one typically develops an expertise in a particular field and gets to know the language of that field. Particularly important in fatty acids. There's so many of them. They all have different names.
Yeah.
It's not just cholesterol, that's
Right.
So Nathan and Jason have been very, very helpful, and they're doing a great job for us. So quick summary. Here's 8 of the papers we've published so far. There's many more in the hopper. And there's 2 that are highlighted in red I wanted to go through specifically. But we have looked at the relationship between genetics and red blood cell fatty acid patterns in one of our cohorts, WIMS. The question is, can you actually predict what fatty acid levels are in the blood from a genetic analysis? Not very well, as a matter of fact. As it turns out, but it's always interesting to know what genes, what particular mutations are associated with different levels of fatty acids. So that's important work. We'll come back to that one next. Dr. McBurney has really led a couple studies on how omega-3s relate to a couple of novel risk markers. One is the red blood cell distribution width, which is a common metric that's used in hematology, but nobody pays a lot of attention to it. But it's turned out to be pretty predictive of outcomes. Having a high distribution width is a bad thing. Neutrophil lymphocyte ratio is a couple of different kinds of white blood cells, and the ratio of those two, one of them relates to acute inflammation, the other is more immunity related. And again, that NLR, neutrophil lymphocyte ratio, is a high level of that is predictive of bad outcomes. And so we've shown in a couple of cohorts now that high omega-3 is associated with better, healthier levels of both of those markers.
Yeah, that's interesting.
It is.
You guys have started to really look at what does having more omega-3s in the red blood cell, which is what we always talk about. We've always kind of assumed it changes how the red blood cell membrane actually functions, or the red blood cell. And so,
Yeah.
and
One of our studies is actually addressing that question. Is there higher omega-3 level in a red blood cell change its biology?
Yeah.
And everybody's always I've thought that, it makes the membrane more fluid. Well, it turns out it doesn't.
Yeah.
We did some pretty high-end experiments in a small group of people, but it was still a very clear outcome. Dr. Schuhart is going to be presenting that data in July of 2023 here next month at ISFAL.
And ISFAL is the
International Society for the Study of Fatty Acids and Lipids, meeting in Nantes, France. It meets every 2 years unless COVID interrupts. And so we'll be presenting lots of papers. FARI will be well represented at that meeting. And one of those papers will be JP's talk about red blood cells.
And fun fact, there was supposed to be an ISFAL meeting in China in 2020. And we were just about to order our plane tickets. We're like, maybe this isn't going to happen.
And as it turned out, it did not happen.
It did not happen. So
skip the year.
Next.
We're back on cycle now, right?
Yeah.
So the variety of things that your number 6 there is your paper, Kristina, looking at how blood DHA levels from different published studies correlate with each other, and particularly in pregnancy. And very helpful, very enlightening paper, I thought, that allows you to compare how well people complied with the treatment in those different studies and how that relates to the outcome.
Yeah, and different labs get different numbers.
Yes, they do.
So they're not on the same scale. And so you can't really judge the blood response unless they're on the same scale. So that was that's part of a future endeavor as well, trying to
harmonize
answers. Yeah.
The number 7, I'll talk about in detail. I'll skip it now, but Dr. McBurney also did his paper looking at a fatty acid fingerprint, meaning looking at not just the red blood cells and not just the fatty acid, the omega-3, at all the fatty acids in a red blood cell and asking whether they would predict risk for all-cause mortality. And we did find that 2 or 3, 4 fatty acids together, including omega-3, were pretty helpful in predicting that outcome. So there's information in red blood cell fatty acid patterns, is the point, that needs to be mined and brought into public use. So let me just do a few slides on a couple of papers. This one here, I think, is particularly timely. The study we looked at in the UK Biobank. Again, that's the group where we don't have direct omega-3 index measures, but we have done studies to correlate the omega-3 level with the plasma marker they use. So the point of this study, of course, was to compare the risk for COVID outcomes, which is 3 of them. One of them is testing positive for COVID. Another one is being hospitalized with COVID, and death at least with COVID, if not from COVID. Nobody's quite sure which it is. And how do they relate to plasma DHA levels? And so that's one of the metrics that's available in the NMR-based suite of analytes from the UK Biobank. So that was our surrogate measure of omega-3 status. And so the study, we looked at events of these COVID-related events that started between January 2021, and we had data through March of 2020 to March of 2021. So not very long, but we had enough data to find interesting relationships. So here's what we reported. And this was published in the American Journal of Clinical Nutrition this year.
Also, real quick, so that time frame is pre-vaccines, right? Widespread vaccine.
Right. Vaccine, there were around 3% of people have been vaccinated by that time.
Right.
In Great Britain, which is where this all is, right?
Yeah.
And so here is outcome 1. In each case, we're looking at 5 columns. These are 5 quintiles of plasma DHA levels. So people in the highest DHA, and this is the top 20%, this is the bottom 20%. And so each one is 20, 40, 60, 80, 100. And then here we're looking at what percent of people in that quintile tested positive for COVID. And as you can see, using the first quintile as sort of the reference, the lowest DHA level, people with higher and higher and higher DHA levels were less and less and less likely to test positive for COVID, which is interesting out of itself. And it's not something we would have perhaps guessed, because we can imagine that omega-3 might help prevent the inflammatory burst, the hyper-inflammatory response, but not necessarily keep you from actually getting the virus. But that's what we observed. And we did that in 26,000 people. That was pretty robust. In this cohort of 110,000 people that were tested, that we have blood levels on, and remember, the blood levels were taken about 10 years ago. And so we're presuming that omega-3 levels in the blood are staying stable over that time. And at the time that COVID hit, had we had blood, would have been wonderful, but we didn't. So we're again assuming that the levels that they had when they came into the UK Biobank study in the first decade of the 21st century are the same. Being hospitalized, same story. The risk for being hospitalized was reduced significantly with the higher DHA levels. Now, again, there's only 838 people that were hospitalized in that period of time. So it was early in COVID.
And in that amount of people who had fatty acids.
And out of the 110,000, right?
Yeah, right.
Right.
Had fatty acid testing, not fatty acids, period.
Yeah, right. We're tested for fatty acids.
Yeah.
Died with COVID. We saw the same thing. Again, there's only 235 people total here that died. So we certainly with the first 4 quintiles, there was a very clear stair step, lower risk until you get to the 5th quintile, then it bumped back up, which we don't understand.
Yeah.
But you publish.
That's real.
That's the deal. Research doesn't always pan out, and you can't explain everything. but this is what we saw. And just to get a sense, so again, we're using plasma DHA percent total fatty acids from the NMR analysis. How does that correlate with the omega-3 index? So this lowest quintile, we've calculated here, the lowest quintile has an estimated omega-3 index, that's a little E, O3I, an estimated omega-3 index of 3.5%. And the top quintile has an estimated level of about 8%, which is a strong confirmation of the 8% target that we've been using for the last almost 20 years as being a healthy target. And so
the less than 4 is
unless
a lot of weight and a lot of, I mean, there's usually a nice stair step that's more of a spectrum. But being less than 4 has a lot of negative consequences usually.
Yeah. And this supports that as well.
Right.
Right. So that's what we did there. The other study I wanted to highlight was one by Dr. Alish Salavila from Barcelona. Here we're looking at red blood cell DHA levels as they might predict a risk for incident of developing dementia or Alzheimer's disease. And this was in Framingham. And so we had 1,490 dementia-free subjects who were over age 65. We decided to look at just people over 65 who didn't have dementia, and we had red blood cells. Their average age at the time was 73 years old. This group, again, you couldn't be under 65, so that's their average age of the group. And we followed them for about 7.3 years for developing dementia or Alzheimer's disease. Hypothesis was, of course, people who had the highest omega-3 levels, DHA levels particularly, would have lower risk for dementia. And as it turned out, we just look at this again. This is instead of using bars, now we're just writing quintile 1 to quintile 5. So this is the quintiles of DHA levels in the red blood cell. And people in the lowest quintile of the reference group. And so if we look at developing Alzheimer's disease, hazard ratio is the risk, it's kind of a relative risk value. So it drops down to people in the highest quintile had 0.51, so roughly a 50% lower risk for developing dementia or Alzheimer's disease over that 7-year period compared to people who had the lowest DHA levels.
And these quintiles, it's a less broad range of blood levels than the other one. Like it's basically going from 4 to 6 on where the cutoffs start. So you're seeing a spectrum.
Well, but yeah, and this is right. This is DHA. It's not the omega-3 index.
Oh, it's just DHA. That's right. Okay. So it is pretty similar.
Pretty similar. So the median value of DHA in red cell, and DHA is the majority of what the omega-3 index is, but EPA adds to it too. But 7% DHA would correlate with at least an 8% omega-3 index.
Yeah.
Yeah. So well, again, nice. Confirmation of that. And here's just another way of looking at the findings in this study. I'll move this over here. We look at the lowest quintile, people who had the lowest omega-3 levels, and the highest quintile, the blue. So this is the rate at which people developed Alzheimer's disease or dementia over a follow-up period. So you can see
every time it steps up is someone who's gotten the diagnosis.
Someone's got a diagnosis, right? And so it steps up year by year by year. But you can see it's obviously a slower development of Alzheimer's disease in the people that had the highest DHA levels. Yeah. So that fit nicely.
Pretty good deal.
Pretty good deal.
Any reason why you looked at EPA too, I assume?
Yeah, we looked at EPA, we looked at omega-3 index, and it was not, EPA was pretty flat, didn't really relate to outcomes. So when you add the no effect of EPA to the changing effect of DHA, it kind of made the omega-3 index per se not as predictive as DHA was alone.
Yeah.
So again, we're not wedded to the omega-3 index. We're studying fatty acids, right?
Yeah. And you guys, did you look at you looked at just the EPA, DHA, omega-3 index, or did you spread it out to multiple
We
fatty
didn't look at,
acids.
we didn't look at omega-6 or
Okay,
trans or saturated or
yeah,
any of
it
that.
just gets too
Too
wide
much. Gets to be
sometimes.
too much. Yeah, right.
So usually when you're proposing these studies, you're picking what group of fatty acids you want to look at, unless it's some of them you guys have done a full spectrum, looking at a fingerprint, trying to see which fatty acids are popping up as the
Yeah.
most addictive. And sometimes you say, I think there's a reason for omega-3s to be related to this, or omega-6s, or
Right.
de novo lipogenesis, which is more saturates and monos in the 16 and 18 range. So it's a little bit of both, a little bit of not having a hypothesis and having a hypothesis.
Yeah, right. It's one sort of open look, just
Yeah.
trying to look for relationships. Yeah, sometimes you have a hypothesis.
But the level of evidence you need for statistical significance, if you don't have a hypothesis, is a lot higher. Like the
It can be, yeah, it depends on how many.
if you have
how
more
many
variables, p-value has
looks.
to be a lot lower. It has to be a lot clearer signal,
Yeah, right.
looking at a ton of stuff.
So some studies are underway. These are not published yet. We're still working on them, but we are, as I mentioned, looking at the effects of omega-3 on red blood cell biology, on blood cell biology. I mentioned red blood cells didn't look to be more fluid or flexible or distensible, but Dr. Schuhart did find that there were, in a variety of white blood cell types, the omega-3s did change their fluidity and functionality, which is interesting because it's the white blood cells that have to squeeze in between into areas of damage. They have stents. Red cells don't. They have to go through capillaries, but they don't have to go through
Damage.
so that's kind of opened up a new world of looking at, well, look what happens to, eosinophil or granulocyte biology with omega-3 fatty acids.
Yeah, because it is representative. When it goes up in the red blood cell, it goes up in basically all the membranes of the white blood cells. It's not gonna, like, it'll in plasma phospholipid also correlates.
Yep.
Yeah.
They correlate. They all correlate. We are ready to resubmit. We've submitted this paper once. It's the FORCE coalition led by Dr. O'Keefe on looking at omega-3 levels in stroke in 20 different cohorts together. That's a big paper. I'll talk about this one in a minute. And then looking at omega-3 status and prediabetes in UK Biobank. We are also interested in how the omega-3 index adds to what's FRS is the Framingham risk score. So it's a way of predicting who's at risk for cardiovascular disease. And so our question is, and in the Framingham risk score, you have about 5 or 6 different things. You have cholesterol, you have HDL cholesterol, you have smoking, you have diabetes, you have age, and you have sex.
Is blood pressure in there? That was like
I think systolic blood pressure is there.
One of them.
Yeah, one of them is. So the question here is, does the omega-3 level add any additional information to that risk score?
Yeah.
And we're doing that in 3 different cohorts, actually. So that's going to be a big project. Dr. Marchioli is in charge of that one.
Wow.
We have submitted a paper on omega-3 levels in all-cause mortality in UK Biobank, submitted to Lancet. It's under review. So that's one. Again, we found the same thing we found in FORCE. It was just nice to confirm it in a UK cohort. Dr. Park in Korea has had an interest in frailty, and so she's looking at how frailty relates to omega-3 levels in the UK Biobank.
Yeah. Cool.
So all good stuff.
Good stuff.
The one I wanted to highlight was a project that Dr. Schuhart is working on in updating the omega-3 world map. This is the map of omega-3 status, published in 2016, so 7 years ago, by Ken Stark and colleagues. And it's
another harmonizing project.
Totally, yeah. Trying to get at omega-3. So there were a lot of countries that were either miscolored, like the US and Canada were classified as under 4% average omega-3 index, and that's not true.
Yeah.
We know that from a lot of
4 to
different.
6.
Many countries are in gray, where it means they've had no data
Mm-hmm.
at times. So we're trying to recolor and update this map.
Yeah.
Countries that were in green, that were meaning average omega-3 index over 8, probably will stay the same. And here Alaska is considered its own country.
And parts of Canada are,
Parts of Canada.
that does make sense, and parts of Russia and,
Yeah, along coastal areas, right?
yeah.
Yeah. What doesn't make sense is Nigeria.
And that's where if you have one small cohort that's representing the country,
Right.
We've seen this, and we've worked with people in Malawi, and there's Lake Malawi, which is huge. And there's a very, people commonly eat a dried fish from Lake Malawi. And when we looked at that group, these women, it was in breast milk, but they had really high DHA levels.
Yeah.
Another group that's in Malawi, but not next to the lake, wouldn't have those levels.
Right.
So
Exactly.
that's where the representative samples, like in the US, it's NHANES. Canada, they have, what is that called?
Well, it's the National Health Survey in Canada.
National Health Survey. So we've worked with Canada. So they've been using our method for a National Health Survey, and it actually gets a representative sample.
Right.
Just
Right.
really,
Most countries have not done representative studies.
no.
Too bad. So anyway, this project is ongoing, and we're working, actually being sponsored by GOED, the global organization for EPA and DHA, because they're interested in how this all looks now, of course. Some other studies pending. We could talk about future studies. There's a lot of that. We have some grants that are pending. NIH, basically, NIH grants. This one here, the first one is something we've applied to. For the Forge Coalition, to try to look at dementia in all these different cohorts. And that's a pending application. Happy to say that this one that you were the PI on, detecting prediabetes with red cell fatty acid profiles, was a funded study now by it's an SBIR, Small Business Innovation Research Grant.
Yeah. So Omega Quant, a for-profit company, can work with a nonprofit. FARI is a nonprofit, or any university or a hospital. That could have been our partner, but it's a grant so that this for-profit, it's really about translating research. And so we have a company that could translate a new fatty acid index very easily, but we need that research done. And before, like I said, Omega Quant, we would just spend time doing it. And then, so now that FARI exists, it's a lot easier for us to apply for funding to cover some of these studies or questions we want to ask that could turn into a commercial product.
Right. I see I've got a typo here.
Yeah.
The second one is supposed to be detecting risk for dementia with the red cell fatty acid profile. This one is also a small business type grant, but it's called an STTR, which is a tech transfer grant, a little bit different than the other one, but we have this application pending at the NIH.
Yeah, this one leans a little bit more on the nonprofit, I think, or it can, so that you get more of the time and funding to work on the biostats, and then it just shifts the focus slightly. But it's still a relationship between a for-profit and nonprofit.
Right. Nathan, our biostatistician, has his own grants, and he's looking at new methods of analyzing fatty acid biomarker studies and simplified methods. So he's working on that. And I think I'll end with some of the companies, foundations that have supported our work, particularly William Donner Foundation. Has been very generous, as has the Tiny Foundation. This is in Canada. St. Luke's Hospital through Dr. O'Keefe has brought some relationships there, as well as actually this company here, this asset management group, is through Dr. O'Keefe's influence as a cardiologist, and they have generously decided to support Dr. O'Keefe's work in FARI. I mentioned GOED is supporting some of our work. Omega Quant Analytics, of course, has been a big supporter of FARI from the beginning. So Omega Quant is still spending money on research, but it's a fixed amount and it goes to FARI. And that makes sense. Pharmavite, one of the major supplement companies, has funded a couple projects, as has physician recommended nutraceuticals, PRN. It's more in the eye omega-3 space. And of course, the NIH is now one of our supporters.
Yeah.
So
Great.
that is the overview of FARI.
It's pretty impressive to see how it's come together in the last 3 years. And I also wanted to mention, it's kind of nice because well, I don't know about how many, but it's an area for you old school researchers who are retired, who can keep using your expertise, but without having to do all the trappings of maybe academic life or industry life. You can just join.
Right.
And
yeah, so I'd say about maybe, well, actually, right, FARI is only paying the salary of one of our scientists, or two of our scientists. Yeah. So, and the rest of us are volunteering.
Yeah.
Some of us are still, so some of these scientists are still active in academics, like Dr. Schuhart. But he loves having access to this data and biostat help.
It opens up his ability to
do
get
other
more
research.
funding. Yeah.
Right.
Yeah. So it's it needs
win-win, as we say.
What did you say?
It's a win-win.
It's a win-win. A win-win. All right. So that's the deal. Nothing else to say about it.
And nothing much else to say. Hope it's been helpful to people. If they have questions, of course, contact us. And if they want to send money our way, we're happy to accept it.
Technically, FARI does have a website.
We have a website called, look up Fatty Acid Research Institute, you'll find it.
Yep.
And there's a big support button that hardly anybody's ever hit, but anyway, it's there.
It's there. Crowdsourced research.
Crowdsourcing, right. Exactly.
Yeah. But cool. Well, thanks for putting that together and talking about it.
We'll sign off and see you next time on Omega Matters. Huh?
All right. Bye.
Bye.